Welcome to Antibody Basics by Biointron, Part 8. In this episode, we'll talk about therapeutics targeting cancer. What is cancer immunotherapy? Cancer immunotherapy leverages the body's immune system to fight cancer more selectively and effectively than traditional methods such as chemotherapy and radiation. There are several types of immunotherapies, including monoclonal antibodies, checkpoint inhibitors, cancer vaccines, CAR T-cell therapy, and oncolytic virus therapies. The road towards cancer immunotherapy began in the 1890s. William Coley, a surgeon, observed spontaneous remission in cancer patients after a bacterial infection. This sparked the concept of harnessing the immune system to fight cancer. Between the 1940s and 1970s, there was renewed interest in the immune system. This time period included major discoveries of interferons, T cells, dendritic cells, and natural killer cell activity. By the 1980s, monoclonal antibodies and CAR T-cell therapies were developed. The development of the techniques for creating targeted antibodies and engineering immune cells opened the doors for novel immunotherapy approaches. In the 1990s to the 2000s, the identification of molecules like CTLA-4 and PD-1 that suppress immune responses led to the development of checkpoint inhibitor drugs. And now, from the 2010s to the present, is the era of new therapies. Current research focuses on tailoring immunotherapy to specific cancers, new modalities, and the combination of technologies such as AI. There are three main target focuses when developing cancer drugs. One of them is targeted delivery. This involves the design of drugs or therapeutic agents that are directed specifically to the tumor site, minimizing exposure to healthy tissues. This precision not only enhances the therapeutic efficacy, but also reduces the side effects typically associated with conventional chemotherapy. For instance, antibody-drug conjugates are where antibodies direct cytotoxic drugs to cancer cells, or we could use liposomal delivery systems. These encapsulate drugs and release them in response to specific tumor microenvironment conditions. Another target is tumor selectivity. High tumor selectivity is critical to maximizing therapeutic success and minimizing harm to normal tissues. Selectivity can be achieved by exploiting the unique biological characteristics of tumor cells, such as overexpressed receptors or mutated proteins not found in healthy cells. One of the most successful examples of tumor selectivity is the use of monoclonal antibodies that target specific antigens or receptors overexpressed on the surface of tumor cells, such as HER2 in breast cancer. We also have stability as a major target, as a drug must remain stable long enough to reach its target without degradation or premature release of its active components. Major challenges include the degradation of drugs by enzymes and immune clearance, so researchers may modify molecular structures, add protective coatings, or use delivery vehicles for the drugs. There are now several successful antibody cancer drugs in the market. Here we have highlighted four. The first, rituximab, was approved in 1997. Targeting CD20 to treat non-Hodgkin lymphoma. It is one of the first monoclonal antibodies to be approved for cancer. Next, we have trastuzumab, which was approved in 1998, targeting HER2 to treat breast cancer. By attaching to the growth factor antigen, it stops the breast cancer cells from growing and dividing. We also have ipilimumab, which was approved in 2011. Targeting CTLA-4 to treat metastatic melanoma. It is the first checkpoint inhibitor approved by the FDA. Similarly, nivolumab was approved in 2014, targeting PD-1 to treat melanoma and non-small cell lung cancer. It is also a checkpoint inhibitor. There are a few challenges associated with cancer immunotherapy development, a major one being resistance issues. Acquired therapeutic resistance against antibodies is a major challenge. This can happen due to a mutation being present in the tumor before the start of the treatment, continuous immune selection pressure of the treatment causing subsequent changes in signaling of survival pathways, epithelial to mesenchymal transition resulting in loss of cell-to-cell contact and becoming more migratory, and lower or heterogeneous expression of antigens and inhibition of complement-dependent cytotoxicity. We also have the challenge of MHC class I expression loss. Each cancer cell differs from healthy cells around the tumor by over 11,000 genomic mutations. While some may lead to tumor-associated antigens expressed on the cell surface to become drug targets, others may lose their MHC class I expression. The major histocompatibility complex is a group of genes that code for proteins on cell surfaces, helping the immune system to recognize foreign substances. They are necessary for distinguishing between self and non-self cells and for presenting antigens to T cells. Thus, losing the expression hampers the effectiveness of immunotherapies that rely on the recognition of MHC-presented antigens by the immune system. Future directions include ADCs, multispecifics, and personalized cancer treatments. Antibody-drug conjugates are an emerging modality that combine a monoclonal antibody with a cytotoxic payload, allowing them to deliver therapy more efficiently than conventional chemotherapy. Kadcyla is an ADC that improves upon the success of Herceptin, the antibody mentioned previously before, by linking it to a potent cytotoxic drug DM1. Besides ADCs, antibody small interfering RNA conjugates and antibody cytokine fusions are drawing more interest. Multispecific antibodies like bispecifics and trispecifics are useful for cancer as more than one mediator contributes to the activation of cancer-related signaling pathways. In cancer immunotherapy, several mechanisms have been developed. This includes engaging T cells or other immune cells to specifically eliminate the tumor cells, or bridging receptors to block or activate synergistic signaling pathways. We also could target multiple tumor antigens or different antigen epitopes on tumor cells to increase tumor selectivity. The potential future of personalized medicine for cancer may be with therapeutic antibodies. A recent study proposed a screening of a large panel of antibodies with each patient's tumor sample to design initial treatment combinations combining 2-3 antibodies. This would allow killing of the maximum number of tumor cells, in addition to having a backup or additional combination of antibodies with chemotherapeutics or inhibitors. Biointron focuses on antibody discovery, expression, and optimization services. Our main feature of antibody expression allows your gene sequence to become purified antibodies in only 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. And no antibody means no cost. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca per 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also have several other services, including but not limited to bispecific antibody production, afucosylated antibodies, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression in the mammalian system, and CHOK1 stable cell line generation. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.